So the overall flow here, I'll sort of discuss some of the background information, go through some examples from the literature of techniques or methods to help increase sensitivity, and then I'll finish up with some examples that we did at NIST. Becky Hill, my office mate and close colleague, she did a bunch of mixtures with low template sort of DNA protocols. So I'd like to, again, thank the NIJ for funding NIST, and so this is our standard disclaimer I should have presented in my other two talks. But so mixtures from high-quality DNA templates with all alleles above the stochastic threshold. Having an animation problem here. But you're supposed to see the big easy button from Staples. I stole that from Ray Wickenheiser. The reality is we typically have mixtures like this. So you have a major allele at 20, you have a 26 allele, and then you have the 25 allele. And the peak height ratio of this minor component over here, well, is that stutter? It's at 14.7%. It's under 15%. But it's also peak height ratio of 39.4%. So I don't know. Maybe it is stutter. Maybe it's not. The issue here is we're at this low template level. It's difficult to distinguish whether this peak at 25 is an authentic stutter peak or if it's an allele. So in the definitions in the SWGDAM interpretation guidelines, the definition of a stochastic effect is that you have the observation of intralocus peak imbalance and or allele dropout resulting from random disproportionate amplification of alleles in low quantity template. But wait, I don't do low template testing. I always amplify 1 nanogram of DNA. This is a slide that Robin Cotton presented several years ago at a workshop at the American Academy of Forensic Sciences. And if you consider in an 8-to-1 contribution of two components, in 1 nanogram, that minor component, you're amplifying about 14 cells. And so as you go down, half a nanogram, 0.25 nanograms, the number of cells that you're amplifying from the minor component, you're down below the stochastic threshold. So you are, in fact, amplifying low template DNA in a 1 nanogram sample. So some definitions, there's no real clear definition of what is low template DNA. Typically, you're working with less than 100 to 200 picograms of genomic DNA. Some people define it as data below the stochastic threshold level where amplification is not as reliable. This is typically, you're talking around 150 to 200 RFUs. Having too few copies of DNA template to ensure reliable PCR amplification. And it can often be the minor component of a mixture with low template DNA, as I gave you the example earlier. Of course, this is something that we've already discussed earlier, this what's called straddle data, or the data that's between your analytical threshold and your stochastic threshold. So only one allele in this pair would be above your stochastic threshold. And this can just simply be because, well, maybe DNA degradation or inhibition, or just the fact that you're amplifying a limited number of molecules. But at this low level, the potential for having this type of straddle data is high. So it's best, if you have this situation, to re-amplify and try to get more input DNA. Otherwise, especially if you're considering this as using exclusionary statistics, this would be a locus that you would not be that would not be usable for statistics. So that would be the locus would be inconclusive. Peter Gill and John Buckleton call this the threshold of a dilemma. This is an excellent response here to criticism by Bruce Bedoli et al. And in their response, Gill and Buckleton discussed this, that suppose in this example here, we have obviously a major contributor at 19 and 24. We also have a minor contributor here at 17 and 25. Now, if my stochastic threshold is at 150, we're okay. We can use this locus in exclusionary statistics, and we could do the CPI as normal. But if my stochastic threshold was, well, not even 175, let's just say 160, I can no longer use this for random and non-excluded statistics. It's okay to use for exclusion purposes, and if you're doing likelihood ratios or random match probability, you could still use this because of the fact that you're considering an assumption that there are two people in this mixture. With the exclusionary statistics, you're not assuming the number of contributors. So in this case, your locus has fallen off the cliff. This is from that discussion. So Gill and Buckleton, they discussed this. There's a risk zone. So when you have alleles that are, very high, above, 500, 600, 1,000 RFUs, everything's fine. But once you start to get down into this risk zone, you start to get the probability of dropout is starting to increase here as you drop down. So John may talk more about this later, so I'll let him take control of that. So the scientific reasoning behind the stochastic threshold, and one reason I think, again, I'm not speaking on behalf of the SWGNAM group, but one reason why thresholds were mentioned and discussed at great length in that document is that when you do have stochastic fluctuation, interpreting your data becomes problematic. Because of the fact you can have allele dropout, you can have your mixture ratio can be variable, and of course you're amplifying low template DNA, so you have the chances of increased stutter and all of the other problems that you can have. So the bottom line is that your input levels of DNA should be sufficiently high to try to avoid this situation where you have data that's between your analytical and stochastic thresholds. It's great if you can get everything above stochastic threshold. So you have to interpret mixtures cautiously when you have low-level data, as peak intensities are quite variable. This is an example, again, that Becky has generated in our lab. So some of the issues when you have these stochastic or random effects with low template DNA. Your peak height imbalance can be very great. So here we have 30% peak height ratio. This is identifier. This is 30 picograms and 31 cycles. So these are using higher sensitivity techniques and amplification using more cycles and additional TAC. But you can see that when you're down at 30 picograms, you're down at this low-level input, you can get these types of drastic effects, such as exaggerated here peak height ratio. You can also get dropout, where the 14 allele has completely dropped out of this particular example here. So the consequence is that you're losing true signal. So it's potential to get a false negative. The other possibility is that you could gain a false signal. So again, I showed this example earlier. This is a 10 picograms, 31 cycles, the exaggerated stutter here, 64%. So is this a single source sample, or is this a mixture of two different people or three different people? So it's this can confound interpretation. You can also get what's called allelic drop-in, which is sporadic potential contamination. So here in this case, in this example here on the right, far right, VWA, 10 picograms, 31 cycles. It's an 18-19, but this 16 allele popped up. So it's not in a stutter position, which often you'll see drop-ins. Most of the time can be explained by potentially exaggerated stutter. So this may potentially be some low-level contaminant that was present in the plasticware or the buffer system or whatever that's being amplified. It tends to be very random, very sporadic, and usually not repeatable. So what are some of the targets, or what are some of the ways that you can improve DNA quantity and increase sensitivity? So there's this paper that Peter Gill, and Jim Curran, and Elliott published in Nucleic Acids Research in 2005. It's a really, really nice paper. It's a graphical simulation model for the entire DNA process associated with STRs. So basically, they are looking at every step during the STR process, and looking at regions or areas where you could get problems. And so, you know, from the very start, when you collect the sample, you're only collecting N number of cells. Often you're not collecting the entire complement of DNA from the stain. So you're only taking a subset of DNA from the stain to begin with on your swab. And then when you go to extract, the extraction efficiency is definitely not 100%. So of that portion that you collected, the big N, you get a little n of how much target DNA or copies survived the extraction process. Of course, we're all familiar with the double swabbing technique, and Todd likes to point this out. You know, this was a technique that was developed to get saliva off of skin cells, and we tend to use it for every other type of collection method now. So it's certainly a classic method to collect DNA. I just lost my animation. Okay, well, so how do you measure extraction efficiency? There are multiple ways of doing this. First of all, some people in the literature, if you take a look at the literature, how many loci are successfully genotyped? This paper here is looking at DNA off of buccal swabs on FTA paper. They used a pass-fail system as to whether it worked or not. This is a paper from, I think from Katie Horseman Hall, Virginia, looking at the number of STR profiles. So you're looking at a weak partial, a strong partial, a full profile, or you have nothing. So full profile, partial profile, or no profile. So there are ways that you can determine the efficiency of extraction based upon what alleles are present. Another way that you can also look at it is just to simply say, I have 5 nanograms of DNA I put here on the podium. I swab that. How many nanograms do I get out of that without doing the PCR, counting alleles? And so there's several papers. This is from FSI Genetics supplemental series in 2009, looking at recovery of semen from cotton swabs, and they are showing here that you're getting less than 30% recovery. Here's another paper from JFS looking at about 33% recovery by putting known amounts of DNA and then extracting. This is an experiment that we did at NIST where we took 100 microliters of epithelial cells. So these are our cell lines, and so we did cell counting. We have a good idea of the number of cells. put them in 100 microliters of solution in a Teflon tube, and then swab that with a buccal swab to sort of simulate, swabbing from inside of the cheek. And what we found is that you get on average about 16 to 21% recovery of the DNA that you put into the Teflon tube. So why does this matter? Well, because of the low extraction efficiency, you could actually lower the sample quantity that you have and push yourself into the low template range. So if you have 1 nanogram of DNA that you're trying to collect, and after extraction you've lost 70 to 80% of it, well, now you're at 200 to 300 picograms of DNA, and that may be enough, but if you have less than 1 nanogram, and you're down now into, say, 50 picograms to 100 picograms, then there's quite a bit of DNA that you've left behind. And now you're into the low template DNA range. Moving back to this Peter Gill paper. So once you've gone through extraction, the next step is to take out a sample from your extract to go on to PCR. And so you are removing X number of templates out of your tube. So this can be simulated by using a binomial model. So just to show as an example, you have red alleles and blue alleles, and this may be what's in your extract, but when you put your pipette in there to pull out template, you may only pull out, say, 3 alleles that are red and 1 that's blue, so you'll get this imbalance when you do PCR. Or by chance, again, using a binomial distribution, you may only pull out 2 of the blue alleles, so you've completely lost the red, so you've completely lost your sister allele here. So PCR, again, is not a 100% efficient process all the time. And it's certainly, if you can approach 100% PCR amplification or PCR efficiency, then that's great. But oftentimes, especially if you have inhibition or degradation, then your PCR efficiency will be lower. So you may not get sufficient amplification. So in other words, you may have pulled out 3 alleles and 1 allele, but by the random PCR process, you may only amplify the 3, but the 1 gets amplified, but it doesn't get amplified enough to produce a signal. So you have to be able to not just amplify it, but you have to amplify it sufficiently so that it creates a little peak on your electrophoregram. So at 28 cycles, the peak may be here below your noise, so to speak, so it doesn't cross that threshold to where you visualize it. And then the final thing is that you've done your PCR and you've amplified, and now you're ready to put it on the CE. So electrophoresis, again, is a place where you can lose information. Perhaps not enough amplicons get into the CE, get injected into the capillary. There are different procedures or ways to increase sensitivity. Bruce Badoli has an article in the Croatian Medical Journal. He lists a number of examples for procedures to increase sensitivity. I'll go through a few of these and show you some of the advantages and disadvantages. One way is to reduce the PCR volume. So instead of doing a 50 microliter PCR, you do a 5 microliter PCR. The advantage is that you can concentrate your PCR product. The disadvantage is that you can also concentrate inhibitors, and you can, of course, pipetting errors. If you're only pipetting 5 microliters of volume, you can get errors there. And you can also decrease the volume of template that you're adding. So instead of adding 10 microliters of DNA, you may only add 1 microliter of DNA. So this is an example from Benoit Leclerc in 2003, where they were looking at here 40 microliter PCR. I know this is kind of hard to see, but the scale here is the same, and what they were trying to do is to visualize the minor component. You see these arrows here. So at 40 microliters, 2 nanograms total, 40 microliters of PCR amplification, you get a number of these minor alleles are present. If you do a 5 microliter PCR with a half a nanogram of DNA, so you've concentrated it down, or you've concentrated the from 40 down to 5, you can see that these peaks are a little bit higher than what you get with 40 microliters of PCR. So this far right peak is a little bit higher than this peak here. So by sort of crowding the PCR space, you can potentially increase the successful amplification. Whole genome amplification, of course, is a technique that has, for a very long time, has been mentioned as a way to increase the quantity of DNA template before you do PCR. So you simply add these enzymes that will randomly amplify genomic DNA, usually at room temperature. So you just sits on the bench for a day or so, and then you can go in and you started off with, say, picogram quantities. You've now produced microgram quantities of DNA by this random amplification process. The disadvantage is that it amplifies all of the DNA. So if you have any bacterial DNA in there, it gets amplified too. So it's not human specific. And it can also, this technique can suffer from preferential amplification. So maybe one chromosome gets amplified at a much greater rate than the other chromosome, and so you can get these major differences in peak height ratios. Jack Ballantyne has published a paper on this technique in analytical biochemistry. Again, just to show you, this is 5 picograms of vaginal epithelial cells, and you can see that at D3 and FGA, D21, D13, you're getting this peak height imbalance. It could potentially be a product of just the fact that you have 5 picograms, but notice the RFU values here are quite high. So again, the technique is can suffer from the peak height ratio issue here. This is the paper I mentioned earlier today. This is a group from the Netherlands, Ate Kloosterman's group. And this is a paper that they published in JFS in 2009 by looking at increasing the injection time or the voltage time for your capillary. And so the advantages here is that by increasing the time and the voltage amount, you can put more amplicons into the capillary, so therefore you can increase signal. The disadvantages, though, is that you can also increase your analytical threshold and your stochastic threshold. So just to show you a couple of examples here from their paper, these are 3 separate control DNAs, and they're measuring, this is using SGM+, and they're measuring the gain, the percent of alleles that are amplified, or the percent of alleles that are visualized by increasing the voltage and the injection time. So this, for example, here is 16 picograms. Under their standard injection, they only recover about 35% of the alleles, but by increasing injection to 9 volts, increasing voltage to 9 volts, they get 90% of the alleles present there. So this was consistent among their 3 different controls, that even at very low level amplification down to 16 and 8 picograms, they're able to recover more alleles by increasing the injection. So this is another example. They looked at 4 different techniques here. This is 3 kilovolts, their standard, 9 kilovolts, the extra. This is amplifying at 34 cycles, and this is amplifying at 28 cycles. And then this is from a paper that Ate Kloosterman published a long time ago. So you amplify 28 cycles identifier, then after the end of 28 cycles, you pop the top, you add more tack, you put it back in for 6 more cycles. And so they found that the increased injection time performed just as well as increased cycles in the samples they were looking at these low levels, 16 and 8 picograms. And this is just showing a sort of, it's very difficult to see, but I recommend if you take a look at this paper, you can get a better view of this figure, but this is showing the gain and the alleles. Now, of course, we talked about if you change your injection times and so forth, you're going to have to reevaluate your changes in threshold. So this is on the 3500, looking at, on the bottom here, 10-second injection. The highest peak that we observed in the PET red channel was 37 RFUs. But if you look at adding additional 5 seconds, which is the default on the 3500, your highest peak is at 50 RFUs. So you're increasing your analytical threshold too. So sometimes the gain may not be that great, because not only are you raising, of course, the signal, but you're also increasing your thresholds too. Post-PCR removal of salts. So, of course, all of the components of PCR, like magnesium and potassium and all of the ions that are there, is necessary for PCR amplification, but not necessary, and actually not that great, when it comes time for electrophoresis. So these charged ions will compete with DNA to get into the capillary. And so one way that you can overcome this is to use a column, filtration column, that will filtrate out or remove a lot of the ions, and therefore you have less competition between the amplicons, the PCR amplicons, and the ions. The disadvantages is that, of course, it will increase your stochastic threshold too, and it's also an added cost. So these little filtration cartridges are expensive. It also adds time to the process too. Jack, once again, very prolific Jack, published in JFS in 2007. He looked at this technique for improving, low copy number DNA analysis by this post-PCR purification. So this is the fold increase, looking at 3 different columns. So he's got the minolute, the Y50 from Microcon, and the Microcon montage. So each one of these gave, we'll just take a look here at, say, 78 picograms of input DNA. He's able to get a 4 to 6-fold increase in the signal, from just putting it on regular versus doing this filtration device here. So it can greatly increase the signal. And this is from a table in Jack's paper here. So, 39 picograms, 20 picograms. If you're unpurified, you're only getting, a few of the alleles present. But if you do this filtration, you're able to, again, remove those ions from competition and getting more amplicons into the capillary. This is some work that Becky did. We looked at minolute, and again, it does a great job. Look at the increase from on top where you have no minolute to on the bottom here, 68, 70% increase in signal compared to no minolute. But some of the disadvantages Jack pointed out was that your percent stutter increases. So yeah, you're putting more amplicons into the capillary. That includes stutter peaks too. So this is the red arrow is showing that this is the stutter that you're putting in regularly, and this is when you inject. You're also increasing the percent stutter in your system too. This is another slide from Becky. You're starting to see pull up here from the size standard that you would not see normally under your normal injection. So again, this is raising your threshold too. So it may be a moot point in that you wouldn't count those or you wouldn't consider those because they would be below your analytical. So there may not be much of a gain there. Nested PCR, this is not really used. This is just more of an academic process. But the advantage is that you can, again, increase the number of target amplicons, but you also can increase your probability of contamination. So basically the process of nested PCR is that you would do 30 cycles of PCR, and if you take that 30 cycles and put it on a capillary, you get no result. But if you take this PCR product and use that in another round of PCR using primers that are internal to your first round primers, you do this for 25 cycles, then you would get an additional number of amplicons that can give success. This is in this paper they used it in a case where they're trying to identify an individual. They have the mother and father alleles here, and they were trying to identify from the evidence the son. So they were able to get it. No result with the first rounds of PCR, but were able to get with nested PCR the results. But of course, that method suffers greatly from potential for contamination. Okay, and so finally, we'll talk about the one major way to increase sensitivity, of course, is to increase cycles, additional TAC, or improved buffers. Of course, this will increase the number of target amplicons, but the disadvantage is that you can increase your risk of contamination. You may have to increase thresholds, and of course, you're looking at stochastic effects. So Peter Gill and the Forensic Science Service pioneered this technique back in 2000. Classic paper in Forensic Science International. There's been a lot of work on low template DNA testing. Van Oorschot published in Nature DNA fingerprints from fingerprints, so from touch DNA. And of course, the FSS led the way in this technique. One thing we have to be concerned with, or at least aware of, is that these new STR kits have modified buffer formulations and polymerases that are already part of the master mix. So it may not be, well, I'm not adding extra TAC, but again, with his proprietary, we don't really know what the formulation is. There may be extra TAC in the buffer. So you have to be aware of this, and you also have to do your proper validation studies to know your limits. These kits tend to be way more sensitive compared to those that are currently used by most forensic labs, and the kits can also contain an extra cycle or two as part of their standard method. So this is just looking at one particular kit, PowerPlex 16 Hot Start. This is 1 nanogram input at 30 cycles. Very nice. This is a complete, fully heterozygous sample. Nice balance. Chris McGuire and I, we were chatting yesterday and over a cool beverage. It was, Chris had tea and I had lemonade just to make. And Chris was impressed. This is the first time he's looked at the Identifier Plus, and he was remarking that, my gosh, the peak height balances are just phenomenal here. So the kits are very nice and well balanced with 1 nanogram type inputs. But once you start getting less and less DNA, of course, you start to get the imbalance. So this is 30 picograms, so you can start to see imbalance at several of the loci. And here at 10 picograms, 34 cycles, you're starting to get allelic dropout and complete locus dropout here of penta D. So again, this was the first paper, at least from the ancient DNA field, Pierre Taberlet. They were trying to develop a technique where they could amplify very low quantity of DNA to try to get nuclear STR DNA. You know, a lot of ancient DNA studies focus on mito, and so here they were looking at trying to get STR data, and they were using 7 replicates and, you know, to account for potential drop in and drop out. And so Peter Gill, his paper came out in 2000, and they recommended duplication of observed alleles and replicates to help in this process. So again, 10 picograms, this is a cautionary note. I'll talk about this later. Here at D3, 14 allele, you know, we're talking 1,500, nearly 1,500 RFUs. No hint of a little blip. So this could probably be a homozygote, but it's not. So this is the Peter Gill paper, and again, what they recommended doing in that paper is to take multiple replicates. So instead of just doing one amplification and going off of that, the recommendation is to do multiple amplifications and then develop a consensus profile where alleles that are observed at least twice you would score. And so you can see here at D21, the control sample here is a 2832.2. They were able to observe that amplification of those 2 alleles twice, so they're confident that that's the true call there. But like, for example, in FGA, which was a 2023, they only observed the 20 allele twice. And so they gave the F designation there as there's a potential that there may be another allele. We're not confident 100% that this is a homozygote, but we'll give this F designation as it was perhaps a failure to amplify the sister allele. In this one case here, 6 replicates, they did see one case here in this box of an 18 allele, which is a drop-in. Most likely, perhaps, a stutter. So it was probably in the first few rounds of PCR, there was an 18 allele from the stutter of the 19 that got amplified in much greater efficiency than the 19 allele. So therefore, the 18 is showing up. Or it could just be a random contaminant. But it was only seen once, so it wasn't repeated. So the fact that they only saw the 19 twice and the 18 once, they said the 19 we're fairly confident in, but there may be another allele there, but we'll put the F designation there. So the New York OCME, which is using this type of low template testing, uses the Z designation instead of F, just in case you see that in their publications. So the suggestions for doing optimal results with low template testing is that typically you would produce 2 or 3 amplicons and develop a consensus sequence. And you could only score an allele as if you saw it twice, at least twice. It's also important that you use extremely sterile environment for your setting up your PCR. I mean, you're almost using mitochondrial DNA techniques because of the potential for contamination. So your laboratory has to be set up in such a way that you could process this type of evidence. So you take the extract, you would split the extract into 3 separate PCR reactions, you develop a profile, and then you come up with a consensus. Now remember this cautionary note. This again was the 19, where the 19 allele had dropped out. This is my 14, it's almost 1,500 RFUs. This is data that Becky generated. Here's 4 additional replicates. So we have 5 replicates here. If I were to pick 3 of those, any 3 at random, I would still get the correct genotype. So just doing one amplification would give potentially an incorrect genotype. I would have potentially only typed the 19 here, or just the 14. But if I were to take any 3 of these together and develop a consensus, I would come up with the correct genotype. So if you're looking at just one amplification, there is a chance, because you're amplifying low-level DNA, there's a potential that you can get an unreliable result. But if you do the replicates and come up with a consensus, so for example, you would do 2 replicates, come up with a consensus, and then you would have the third one as sort of a standby if you need it, and you develop your consensus profile using the interpretational rules based upon your validation. In that issue of Croatian Medical Journal that I mentioned earlier that Bruce Badoli published his article, Teresa Kerrigan from New York OCME published their validation of their low template DNA testing. That journal is free, so it's online. If you go to the Croatian Medical Journal, I think it was from June of 2009, I think. You can download those articles. So, as I mentioned earlier, Becky Hill did some low template DNA mixture examples in our lab, just to sort of get an idea of how would it work with doing low template mixture analysis. So she did a couple of different experiments. First of all, 2 samples, male and female, mixed together in different ratios, 1:3 and 1:5, at 2 different DNA inputs, 1 nanogram or 100 picograms. She was only doing the 1:5, or 50 picograms, 1:3. So 2 different ratios and 2 different concentrations. She also did a 3-person mixture with 2 males and a female mixed together in a 1:2:3 ratio using 1 nanogram or 100 picograms. So we looked at identifier plus, normal, 28 cycles, and 31 cycles. We do half reactions in our lab. Did 5 replicates, and then using 3 extra cycles. And so we looked at the minor contributors that were called with the 3 extra cycles. So here's an example. This is the male. Here's the female. This is 1 nanogram. And then this is the mixture, the 2-person mixture at 28 cycles. So looks like a mixture. However, at 100 picograms, 2-person mixture at a 1:5 ratio, again, 31 cycles here, you can see that between any 2 examples, notice the peak heights can be very variable. So the peak height ratio between these 2 peaks here are a little bit different here than on top. And even look at this one, so much lower. So you're getting a lot of variation in the peak height ratio between any 2 examples. So look at the first top left. So notice this 13 peak is much lower than the 12 and 14, but in here, the 13 peak is almost the same as the 12. So again, just taking one shot, one example, one amplification, you can get highly variable results. So it's important that you do this type of consensus testing. And the FSS has recently published a paper where they've looked at low template DNA mixtures, and based upon using their training and their highly qualified analysts, they were able to correctly identify a great deal of these mixtures. Notice here, this allele here, the 30 allele, has dropped out. So it's not present at all. So this is just making the point again. Your peak height ratio is no longer the same, and as is your mix ratio. It no longer follows a 1:5 in that case. This is looking at the 3-person mixture. Again, these are the controls. So here's a male, female, another male. And so this is what you would get at 28 cycles. Notice 6 alleles here at D7. Okay, so it's a 3-person mixture. But again, looking at the different amplifications, you can get a lot of variation in the peak heights, say, between this amplification here and the peak heights here. And you can get dropout. So you are at the mercy of all of those things that can happen when you're down at the stochastic range, at the low stochastic levels. This is the paper I was talking about that's just in press in FSI Genetics by John Wetton and all, Jillian Tully, Jonathan Whittaker. So you're talking, a lot of really experienced people part of this paper, and they were able to do a successful job of identifying a major minor or a major consensus at different ratios, looking at these replicates of mixtures. So to summarize, there are multiple methods that you can use to increase sensitivity with low-level mixtures. As John likes to call them, enhanced interrogation techniques need to be, fully validated before you consider this. So it's one thing just to say, well, why don't we just inject it for a few more, kilovolts or whatever, or why don't we just add another cycle or two. Make sure that you've validated that first, because it may cause a change in your thresholds and your interpretational guidelines. But really, I think a huge focus on improving the collection, extraction, and reamplification is a way to sort of increase the amount of starting DNA so that you may not have to go to these, post-PCR cleaning up and extra cycles and extra tacks. So it's a way to, hopefully, if we can improve our extraction and collection procedures as a way to increase the amount of DNA that we're testing. Okay, so I'd like to thank Becky for a lot of the data, and Robin, and of course Peter Gill, who's just recently moved from Strathclyde.